I want to thank Quanterix. It's a big honor to be representing the Huntington's disease field, which doesn't get much word in these kind of conferences, but actually ultrasensitive technologies are making a huge impact in our field, particularly for clinical trials at the minute. And I'm going to give you a whistle-stop tour through the work I've done in my PhD. And I don't have any disclosure slides, but I have to disclose I'm not quite a doctor yet, even though it says on the program, inspired by our talk, so fraud is not our trade secret. So it's a really, really exciting time in Huntington's disease. In 2017, in December, there was a storm of these kind of press release of the headline results of the first trial of the first drug targeting the Huntington protein itself, the root cause of the disease, and trying to lower it within the CNS. And Huntington's disease is this is probably the biggest thing that's happened since we found the gene back in 1993, which is around the same time I think MS had its first disease-modifying therapies. And we have this mutant Huntington protein that does all the damage in HD and causes the brain cell death. And it's caused by this expansion of CAG repeats, for those of you who don't know, over 36, causes the mutated protein that aggregates, and we have this inverse relationship with disease onset and severity. But these results, this dose-dependent lowering of Huntington in the CSF was a huge deal for the field. What really showed was a proof of concept that we could actually use a drug to target the root cause of the disease and lower it. And that was the first time to show it in humans. But this was actually a real win for ultrasensitive technologies in biofluid quantification. The paper that first presented this method that they use, and I'm sorry, it's a different technology, the Singlex platform, but it was the first time we were able to actually quantify mutant Huntington in the CSF of Huntington's patients, because it's very, very low concentration. Without this technology, there would have been no target engagement marker for this trial. This is the classic trajectory or spectrum of the natural history of Huntington's disease. We see a gradual decline in functional abilities. At a certain point, we have motor diagnosis, which has historically been how patients with HD are diagnosed, with increasing motor impairment, cognitive impairment. But we also know that there's a lot of early changes in this prodromal stages. And the classic scales that we use, that are still used today as endpoints for clinical trials, is this unified Huntington's disease rating scale. And it has its problems, obviously. It's very biased by clinicians and inter-rater variability, and also huge floor effects. All these scales are only really useful in this overt clinically manifest patient stage. But what I'm really interested in, and I think the field is moving towards, if we do get a disease-modifying therapy, we really want to begin treatment far before any of these symptoms start to manifest, in the hopes of potentially preventing the disease ever occurring. And there's been a huge amount of effort over the last 26 years of observational studies in different cohorts, trying to develop biomarkers and learn as much as we can about the natural history of the disease. But in terms of blood biomarkers, this is kind of the classic result of most of the studies that we've done. They do not, this is not a biomarker of HD progression. And CSF studies have been probably less advanced than other fields, probably because we've had the diagnostic test. Most of the studies had occurred before the genetic test had even or the gene had been discovered. And since then, there's been a huge lack of replicability. A lot of them had very loose connection to the underlying pathology. Again, the sensitivity of technologies was not adequate, and there's been not a lot of validation and results being replicated. until we get to neurofilament light, which I don't need to explain here for this audience, but it's really shook our world in Huntington's disease. It's the biomarker that we've really needed, as we don't need as a diagnostic biomarker as such, but we need something that tells us about the underlying pathology and ongoing neuronal damage. So we looked at it for the first time back in 2017 in the TRACK-HD study, which was a longitudinal multi-site study led by Sarah Tabrisi at UCL. We were kind of blown away by the results. We saw increased neurofilament in all disease groups, and even in this early pre-manifest group, which were people over 10 years from their predicted onset. We saw a really striking relationship with CAG repeat and how neurofilament increases with age. So you see the classic increase in controls, but with increasing CAG repeats, you get an earlier and steeper increase of neurofilament. A single measurement at baseline was able to predict or was associated with those who progressed to motor manifest that were pre-manifest at the beginning of the study. And it had predictive value or prognostic value of subsequent brain atrophy and other clinical decline. So going into, after the study, we knew that Huntington was an important biomarker in terms of tracking disease progression, but also as a pharmacodynamic marker and the disease agent itself. Neurofilament in blood and CSF tracks progression and neuronal damage. But what we don't know or did not know is how these relate to each other, and in combination, what do each of them tell us that the other doesn't, and what are their longitudinal dynamics that will hopefully inform how we interpret their changes in clinical trials. So my PhD was focused around this HD-CSF study led by Ed Wild, who's my boss, and it was an 80-participant study with 20 controls, 20 pre-manifest gene carriers, and 40 early to moderate stage Huntington's disease, and it was designed prospectively to be a 24-month follow-up study. And this is the first of its kind to have very standardized protocol for CSF and matched blood collection with all the clinical, very standard and well-established clinical assessments, as well as this optional MRI component, which allowed the first time to be able to compare these biofluid changes with the more established structural MRI measures. So looking at these in parallel, so we published the baseline results of the study last year. I'm just going to whiz through the headline results. Interestingly, so we have CSF mutant Huntington in green, CSF neurofilament in blue, and plasma neurofilament in red throughout. Neurofilament seems to have a stronger association with all of the clinical scores and the MRI measures. Mutant Huntington was not associated with brain volume, which was surprising to us because it's been showed in mice studies to be released from when you damage with toxic injection into the mice brain. We showed using a short sampling interval of 68 weeks that all three of them had a high stability or within subject stability for all three of the biomarkers. And using this, we were able to do a sample size calculation of using these the lowering of these biomarkers as potential endpoints for clinical trials, and saw that less than 35 participants per arm would be needed at effect sizes as low as 20%. Using a computational method, to try and infer a sequence of events of when these biomarkers change relative to one another, we found that the biofluid biomarkers came out earliest, even before caudate volume, which is one of the earliest structural changes in HD, as well as motor score and the global brain atrophy or brain volume and cognitive changes. So I'm going to go on to present some of the longitudinal data of the 24-month follow-up, which we're getting ready to submit. This is the longitudinal dynamics of all three of the biomarkers, so the raw data. I think the most striking thing here is actually the NFL data when you compare it with the controls. You can see just in gray here, the controls almost sit completely underneath the gene carriers. And we even have the pre-manifest starting to break away from controls at this early stages, which has led me to believe that we're looking at something over kind of 20 years from predicted onset. And we saw a similar CAG-dependent response also with mutant Huntington. And we're using this, so the way I see biomarkers and what my work is continuing to go towards is, can we develop a model for what these biomarkers should look like through the natural history of disease, when they should change from normality, so we can decide on when to start treatments. Using a composite score of the UHDRS components that was developed and is currently being used as the endpoint of the phase 3 Roche trial. We wanted to have a single measure for disease progression, or global disease progression. So with worsening in the x-axis, so the change in CUHDRS, looking at the baseline measurements of all three of the analytes, they were all associated with this subsequent progression. But then comparing it to the rate of change in the analyte, we don't see the same associations apart from neurofilament and plasma. What this the caveat here is that the rate of change of both of the measures, the progression measure and the analytes, are measured with the same time frame. We don't have the capacity in the study to look at, say, the rate of change prior to the subsequent next 2 years, which is why we might not be seeing any association here, but it was surprising for us. And we see a similar thing when we look at it in terms of splitting the groups by how those who progress faster and those who progress slower. So those who progress faster had a higher baseline measurement compared to those who didn't, and there was not as much of a difference in the rate of change in all three analytes. Using receiver operating characteristics, we looked at the ability to distinguish between different disease groups, so controls versus HD mutation carriers. We saw that mutant Huntington, a single measurement at baseline in mutant Huntington, had the superior ability, which was unsurprising as the assay itself does not detect any mutant Huntington in controls. But neurofilament had very high areas under the curve. And when we compare that to the rate of change in the analytes, we see that a single measurement at baseline has greater discriminatory ability. And the same for when we look at pre-manifest versus manifest HD. And what we're trying to do with the data and work that we're building is to try and inform clinical trial design. And one way we thought to do this is by looking at this is a matrix of a single effect size of lowering of these biomarkers, neurofilament in particular, I think it's 20%, and looking at that at a range of participants per arm and length of trial, and we can see the power is the color, to show different ranges of how we could do shorter trials with less participants to make much more efficient studies. So in conclusion, neurofilament and mutant Huntington are a powerful combination in HD. The longitudinal dynamics are clearly distinct from controls, and it seems that baseline neurofilament, I didn't include that slide, but you can read the paper. But a single measurement at baseline seems to be more predictive than the rate of change in the biofluid biomarkers. But the longitudinal dynamics seem to have will probably have their own importance. And we hope that our data will inform or help to model for future trials. So looking forward, there's a lot of things we're looking forward to with the Roche trial and other big players like Wave. But following this, there are various other clinical pharmaceutical companies that are all taking interest in HD with their own Huntington lowering programs. And going forward, we hope that the the optimum aim of these biomarker studies is that we can hopefully, if once we have a disease-modifying treatment, can begin much earlier to to extend the life of these gene carriers that they never get HD. The young adult study, which is led by Sarah Tabrizi, the results of that will be coming out next year, which would hopefully find that exact point where gene carriers go from normal to abnormal and the pathology begins. And where do I see the precision health and HD in the future? So not only can people get their genetic tests, but what's coming out with the genome-wide association studies is that there are other genetic components and SNPs in our genome that modify the onset of disease. And I believe that in the future, someone who's at risk for Huntington's will be able to get tested not only for their CAG repeat, but potentially for other modifiers that influence how their prognostics or their prognosis. And then they will be monitored in a clinic as based on that, potentially getting blood tests on a regular basis, getting their NFL and whatever other blood biomarkers we have then, which may progress to a CSF collection where they look at mutant Huntington, perhaps MRI, and eventually there'll be a decision made between the patient and the doctor together on which disease-modifying therapy to take forward. So in that, I'd like to thank everyone for listening and all my funders, and particularly the Huntington's Disease Society of America, who are continuing to fund me for another 3 years to understand neurofilament light in the full spectrum of Huntington's disease. The only thing now is to fund the neurofilament kits. So if anybody has any good ideas, I'd like them. And I'd like to thank all of our patients and you for listening.